So hi everyone, this is Susie Wood speaking. I'm here to give you a bit of an update on what we've been up to in critical step 1.2.1, which is part of the Enhancing Health and Resilience of New Zealand's Lakes research program. So this project's really all about improving monitoring technologies which can be used to understand water quality and also the biodiversity of New Zealand's lakes. The critical step involves myself, David Hamilton, Chris McBride and Maritz Lehman. So there's sort of three key parts or three key things that we're working on at the moment in this critical step. The first one, which we've largely finished now, has been assessing the use of phycocyanin sensors as a method to measure cyanobacterial biomass in lakes. And a lot of the work has been done by two master's students, Vanessa Codrell, who you see in this picture here, and Caroline Hodge, who finished her master's in 2016. So Vanessa's project has two parts to it. The first one has been really doing some side-by-side comparison of a phycocyanin sensor, which you see Vanessa holding there, and manual or microscope determined cyanobacterial biovolumes. And she's done this over 2 summers at 14 sites in the Rotorua Lakes. So yeah, for each sample she's looked at the species and determined biovolumes, and then she's got a parallel on-site phycocyanin measurement. And for those of you not familiar with phycocyanin, it's a pigment that's very specific to cyanobacteria. So just a very quick overview of some of her results. So really what she's shown is that there is a weak relationship between cyanobacterial biovolumes and phycocyanin as measured by the probe or the sensor. And this varies between lakes and also between years. What we see when we look at a lot of the Rotorua Lakes data is actually that the biovolume over the summers when Vanessa did her work was actually pretty low. And that we know that the sensors below certain thresholds or values actually don't work particularly well, or the relationship's not particularly strong. Even when we had quite high concentrations of cyanobacteria, there was a huge amount of variability. And also you can see from this graph here, there's not very many data points. And there's a whole suite of different reasons why this is. Some of these, or some of the more obvious ones, that are to do with the types of cyanobacteria that we see in these lakes. So when we have large colonial species, the relationships are very weak. The phycocyanin sensors can't penetrate into those colonies, or there's a lot of randomness as to whether a colony floats underneath the sensor when it's taking its reading. Okay, so part 2 of Vanessa's master's project was coming back into the lab and just trying to understand a little bit more about some of these environmental parameters that affect phycocyanin sensors. So she did quite a complex laboratory study which involved one of our key bloom forming species, Microcystis, and she looked at it through the growth cycle and how nitrate, phosphate and light affected both growth rates and phycocyanin sensors. And so Vanessa's just handed in her thesis. And yeah, I won't go into too much detail in the results, but I guess the key thing was that there was lots of variability, and each of these different parameters had a different effect on phycocyanin. And when you think about how that might affect phycocyanin measurements in the field, it really just highlights why there's not a really strong correlation between phycocyanin and these sensor values. So a whole lot of considerations that need to be taken into account if you are going to use these sensors as a proxy for cyanobacterial volume in lake. So really some of the key take-home messages is that they can work well in some lakes, but you really need to validate them and understand what type of species you've got. They do work particularly well or much better generally when cyanobacterial densities are high and you've got species which form quite homogeneous colonies or cell sizes within the lake. So one area why they're not particularly good is an early warning method, so to warn perhaps when you've got a bloom approaching, but one thing they could be really useful for is for issuing on-site warnings. So we know that at the level that under our recreational guidelines you'd issue an alert mode, that actually phycocyanin sensors work quite well. So you potentially could go to a site It probably looks very green anyway, but you could put your probe in and actually get confirmation that actually, this is a really high level of cyanobacteria, and put that health warning sign up right there and then, which could save, a week or several days for the samples to be processed using normal techniques. And yeah, I guess just again to highlight that there are a number of caveats that you need to understand that will affect phycocyanin readings, and these are things like the species composition, the temperature, the light, the nutrients in your lake. And so quite a lot of these experiments, so Caroline Hodge also did a bunch of similar sort of experiments, and her work's been written up into a publication which has been submitted that we hope we will hear about soon. And in more of a summary of these methods and also some of the other methods which are available for monitoring cyanobacteria have also been submitted in a book chapter by myself and David and Michelle Burford. And yeah, I'm sure if you contact any of us, we'd be very happy to pass either of those on to you. Aspect that we're interested in is how can we complement some of the sensors that are already out there on these observational platforms in New Zealand lakes. So there's observational platforms or buoys in about 20 of New Zealand's lakes at the moment. Most of these are measuring things like temperature and dissolved oxygen, pH, conductivity. There's a range of chlorophyll A sensors, phycocyanin sensors, but we're always looking at what's the next thing that we might be able to get on there, which would provide us more useful, more information that will help us understand lake dynamics. And so one of the things that I guess a reasonably recent sensor, or sensors that have been improving dramatically in recent years, are nitrate sensors. And so what we've done to date really was just a little study where we compared the values that we obtained with these sensors versus laboratory determined nitrate values. And we selected a range of lakes which we knew had various different levels of dissolved organic carbon, turbidity, salinity, total suspended solids. And so you can see some of the pictures of the lakes. And we tried to pick a few of the more extreme ones, such as Ellesmere, which we know has quite a high sediment load, slightly saline. Some of the peat lakes in the Waikato region, which we know have quite high total organic carbon. And it was a really useful exercise. We learned quite a lot about how you might go about setting up these nitrate sensors, to perform optimally on your observational platform. So some things such as the total suspended solids had very little effect, but others like salinity really was a big one. And with these new, the sensor that we tried, at least the Trios, it didn't work very well, and actually you need to have the full spectra available. And of course there are sensors, more expensive sensors that will give you that capability. One of the things we learnt that actually, especially when you've got a lake which has got low nitrate levels, if you can set your sensor up so that it's taking multiple measurements but averaging across those, then you just improve the robustness of the data that you obtain. And so yeah, this is still work in progress. I think the next steps now are to look at seeing if we can get some of these out on some buoys and just run them for a few months and see what kind of data we can obtain and what we learn about nitrate concentrations in the lakes. And the ideal would be to get these on some of Chris McBride's new profiling buoys and have a look at them in parallel to the other data that they're obtaining. Sorry, I missed a slide, is using eDNA for assessing lake biodiversity or lake community composition. And so at this point, the aim of this is to try and develop or assess which is the best kind of sampling and analysis methods that we should be using. And ultimately we may want to put these sampling methods out on some of these observational platforms and collect some really cool temporal data. So environmental DNA or environmental metabarcoding it's quite a new area of research. So very simply what we can do is we can go out to the environment, and in our case of course we're working on lakes, so we can take a sample from the lake, and we'll talk a little bit more about how we might do that in the next few slides. We come back to the lab and we can extract the entire DNA of everything that's in that sample. We can then amplify up our gene of interest, and this could be all bacteria or all eukaryotic organisms, or we may choose to target specific groups of organisms such as fish or macroinvertebrates. We then use a technique called high throughput sequencing, which allows us to obtain a huge amount of sequence data very rapidly and simultaneously. So we get something in the order of 10 to 20 million sequences typically in a run. And then it's a whole complex bioinformatics process to go through and assign each of those sequences to reference databases, and provided you've got good reference databases, or there are good databases that you can use internationally, you can then put some taxonomy to each of those sequences. So that's the aim of it. And so what we've done this summer is to do a little bit of a case study, and just to see how effective these methods are and how we can improve and use them. So we've worked on 2 lakes in the Ashburton Lakes area, 2 contrasting lakes. So Lake Emma, which is quite a clear water lake, it's macrophyte dominated, and Lake Maori, which is a very shallow lake, so it's only about half a metre deep. It's really interesting in that it's got this macroalgae bloom that's sitting on the bottom of the lake. At Lake Emma we sampled 5 sites, and at Lake Maori we sampled 3 sites. At each of these lakes we undertook a suite of traditional survey methods, so this included things such as sending Dave out to search around the mud for macroinvertebrates. It involved minnow traps, phyc nets, and characterising macrophyte communities and looking at zooplankton communities, and they were all identified using traditional methods, morphological methods. So in parallel to that, we have assessed 3 different sampling methods. The first is a super simple method in which we just have a falconer tube or a 50 ml tube full of 33 ml of ethanol and then 1.5 ml of sodium acetate. We use a pasta pipette, and we put that just below the surface of the water, and we take 13 ml of straight water, no concentrating, nothing, and we just add that to the tube, mix it up, and keep it cool, and as soon as we can, back in the lab, freeze it. So a super simple method. And at each site we took all of these samples in triplicate. So the other method that we've used is we filtered 15 litres of water. So we take a pump, and we pump water from the middle of the water column. We put this through a 20 micron mesh, and then we refilter that onto a GFC filter, which is cut in half, and then that filter is dropped into a tube which is full of RNA or DNA, or RNA and DNA preservative. So these pictures here just show what these filters look like. So Lake Emma, there was a huge amount of sediment in the water column, so you can see that there, and actually a lot of zooplankton. So all of those tiny little dots you see there are zooplankton. And this is Lake Maori with its lot of macro algae in it. And you can see in some cases, I think this is going to be interesting to see the data because it may be overloaded with macroalgae DNA signals. And the final method, again quite a simple method, which involved dropping a ponograb and taking sediment from the bottom of the lake at each of those sites, just scraping off using sterile methods the very surface of those samples. And so all of that, all of those samples have been analysed back in the lab, sent off to the sequencing facility, and we've just got the results back in the last few weeks, and we're really excited to be analysing those at the moment. And yeah, so far, don't want to give too much away because I want you to listen to my next talk, but the results look really fascinating, and yeah, we're able to characterise a huge amount of diversity from these lakes. So yeah, stay tuned for an update on that. And yeah, I guess ultimately the plan with this is that it's potentially a really rapid method that we could use to characterise lake biodiversity. So like I said, we could put a sampling methodology onto these observational platforms and characterise temporal changes, upscale take samples from many of our 3,000 lakes in New Zealand and really understand a little bit more about the lakes and what are the different communities in them and how are they changing and being affected by all the different pressures that our lakes are under. So yeah, hope you enjoyed that. If you'd like any more information, then please feel free to get in touch, and yeah, I'd love to update you on it in the next talk. Cheers.